just stick to the 20 minutes.
The time's not running, so I can talk as long as what I want, right? Okay.
Thank you.
So I work for CRI, but before this, I was with Delrod for many years. And this is part of research spanning a very long time with many collaborators in different institutes locally and internationally. So just a little bit about what I was working on and what I learned over many years. Okay, biosecurity plays out on a global scale. We know this. It falls into invasion science. An organism which is native in an environment may be transported to a different environment and may become invasive. We call this a biological invasion, and all of our pest arthropods that get transported from somewhere else fall into this category. But in order to become invasive, an organism must first get through a number of different stages, and there are barriers to getting over them. It first needs to actually be transported out of where it is. And this is usually where humans come into play. We transport things all over the world. Even once it's in a new country, it takes time to establish. Not all things that are introduced will establish. Even those that are established in small populations might not spread and might not cause big problems. So over each barrier, this organism needs to come, and we study things along the entire invasion process. So if you're studying the spread of an organism, this is where most people do management of invasion of invasive species or of non-native pests. But work can also be done here on the establishment process, things like early warning systems, containment or eradication of a new pest, and then right in the beginning, preventing a pest coming in the first place. So this is where biosecurity comes in, where pest risk assessments come in to provide that prevention of something coming in. But although we know that prevention has a better cost-benefit ratio than a later management, it's also the most difficult aspect of this invasion process to actually study, and the most difficult to control. And that's particularly true when it's agricultural pests. Why? So why are bugs so problematic when it comes to biosecurity? So when I talk about bugs, I'm talking about insects, mites, nematodes, fungi, snails, viruses, pretty much any other small organism which comes in, insects and mites being the most common. But then also, if we know why they're so problematic, what can we do about it? What I found out over many years is that biosecurity on paper is very well regulated. We have incredibly good international systems, we have trade agreements, the ISPMs are set out really, really well. But in reality, there's severe capacity limitations, and we don't know how well the system's working. Part of this is just that we are trying to still implement tools that we started in the 1960s, 1970s, still trying to do exactly the same thing. But our trade volumes have increased to such an amount that there's no ways we can keep doing the same thing and expect it to work. And this is not just South Africa, this is worldwide. The unknown efficacy is shocking. We don't know because we don't record. It's as simple as that. We might know when there's an interception of a quarantine pest. But how many interceptions of non-quarantine pests are there? How many consignments did we go through before we found that interception? How big is each consignment? All of these are there somewhere, hidden in trade records or shipping manifests, but they're not recorded in one place so that we can actually look and figure out how the system works. Which makes it incredibly difficult to actually say, is our biosecurity working, and should we change it? So I've spent a lot of time trying to do exactly just that. And a big thank you to Delrod while I was working there that they allowed me to use their diagnostic records over a 25-year period. So I looked at over 26,000 samples. These were samples that came to the diagnostics lab. Every single thing was inspected. So I had a full record of exactly what was done. Around 30% of those had contaminants on, very often more than just one. So a contaminant is any organism found on an import, regardless of whether it's phytosanitary or not. We found insects, mites, fungi, bacteria, nematodes, snails, viruses, many, many, many different organisms. If we look at the contaminants, so this is any organism, you'll see very, very clearly fungi, insects, and mites top the list. Significantly more than anything else. But remember now, these are all contaminants. So if we look then only at what's quarantine, the picture changes a little bit, and there we have mites topping the list. So really, truly, our bugs, mites, insects, fungi, are our three biggest problems when it comes to biosecurity, and if we're looking at quarantine, very definitely mites. But why? So back to our little guy with his question, our first why is because they're small. And that seems fairly obvious. If we've got a box of kiwis, on the calyx of the kiwi, those tiny little red dots are a mite. Okay? These are actual interceptions. They're tiny. They're not easy to see in inspections. If you think those are small, look at grape imports. They're sitting between the berry and the stalk are tiny little grapevine bud mites. Okay? They are incredibly difficult to see. So the fact that they are so small and that they hide so much, it makes them really difficult to detect, and so they do slip through quarantine quite easily. Also, because of that, you need well-trained personnel to be able to see them, and you often need specialized equipment. An inspection at the harbor without a microscope is just simply not going to cut it. But what do we do about it? Well, some things seem fairly easy. Firstly, and this seems a little bit counterintuitive, don't rely only on inspections. Biosecurity inspections are meant as a check that the system is working. They are not meant as a biosecurity mitigation measure in and of themselves. And yet far too often we rely on those inspections as if they are the biosecurity. They shouldn't be. Instead, we should be relying on our systems approaches, on our mitigation measures, on making sure orchards are clean, or making sure that our post-harvest measures are up to scratch. There needs to be more investment in training, making sure the personnel know what they're looking for, and then also looking for the right thing using the right method. Like I said, far too often we're told that a visual inspection was conducted and no fungi were seen. How do you see no fungi in a box in a cold store that's only just come into the country? It's not an appropriate detection method. The second why is that insects and mites are incredibly diverse. And here I want to tell you a story of a mite that starts in 2007. And on kiwis from Italy, we intercepted a flat mite, a Brevipulpus species. It was not one of the species present in South Africa. It was similar to Brevipulpus lewisi, which was a quarantine pest at the time, but we couldn't identify it. And because we couldn't identify it, we couldn't be sure whether it's problematic or not. And so we sat. Over the next 16 years, we kept intercepting it. First from Italy, but then also from other countries in Europe. Multiple interceptions, still no answer about what it was. Why? Well, A few reasons. Firstly, we only intercepted adults. So one of the problems there is kiwi then a host? Or was it just a fluke? Secondly, the taxonomy of Brevipulpus is complicated. We weren't sure if it was a new species or if it matched to one of the 300 odd in the genus. And the reason we couldn't figure that out is there's no specimens for comparison. If you intercept something, you only really have what's at hand to compare it to. If it is not something in South Africa, there's nothing to compare it to. Which leads to the third one. Everything is sensitive phytosanitary information and it's confidential. I couldn't ask overseas colleagues. So in this particular case, the leading expert on Brevipulpus works for the US Department of Agriculture in their quarantine section. There's no ways I could have contacted him and said, hey, we just intercepted a brand new thing from Italy. It would have made for a serious breach of confidential information. Luckily, we did get an answer last year. And it came with a roundabout route that there were a few Italian researchers that did a survey on kiwi. They found a new Brevipulpus species. They could examine specimens kept in the Smithsonian, because of course from their side it's not a quarantine issue. And via the other side they contacted me knowing that I work on the genus. So we then do have an answer eventually, 16 years later, but this could have gone so much faster if we had better collaborations and more able to share information with confidentiality agreements in place. I mean, I'm not advocating we just share with everybody, but keeping information so confidential that you cannot get an answer is hamstringing science in biosecurity. So our diversity in insects and mites makes them very difficult to identify. We have a number of cryptic species as well as new species. In my years in the quarantine station, there are 9 Europhyid species that we suspect are new species to science. I cannot describe them, I cannot identify them, because I cannot talk to anybody. And of course, when you don't know what it is, the biology of it and its pest status are unsure, and therefore the biosecurity response is unsure. So what do we do about it? Well, firstly, better international collaboration. Biosecurity plays out on an international stage, and yet, when a problem comes along, we don't want to ask for help internationally. It is needed, along with confidentiality agreements, yes, but scientists share information quite freely as long as they know they're allowed to. There does need to be more emphasis on taxonomic research. We find new things all the time and we do not know what they are. Any risk analysis that gets done needs to also incorporate the possibility of an uncertain threat. You cannot only have it as a black and white. Then my last why, and this is actually the biggest why, and I left it for last, is the fact that they are accidentally transported with agricultural products. Why is this a problem? Well, firstly, the fact that it's accidental. An accidental occurrence is very difficult to predict. Plus, you cannot control it directly. So, for instance, we have regulations against prohibited species being brought into the country. Okay? A fruit fly, for example. You may not import a fruit fly into South Africa. But nobody's importing fruit flies. They're bringing in fruit. And we can't have a ban against fruit. Okay, so you cannot directly control your prohibited organism. You need to control the pathway that it's on. And that pathway is agricultural products. And I think Kofi Annan put it very well when he said that arguing against globalization is like arguing against the laws of gravity. Globalization is going to continue to increase. Trade is going to continue to increase. And as it increases, we need to keep up with this increasing volume. We can't stop it, but we need to learn how to control it so that it is still safe. So our why with the transport and agricultural products, it's accidental, difficult to predict, and our trade will continue to increase. But what do we do about it? Well, first of all, we need to study the pathway. There's a lot of emphasis on studying the organisms, and that works if your organism is intentionally brought in. So if I wanted to import a new ornamental plant, and I did a risk analysis on that ornamental plant, it would help. But I'm not importing a new pest. So although risk assessments do need to be done, they need to be incorporated into a pathway risk assessment rather than purely individual pest assessments. And we need to always remember it's the pathway that is controlled, not the organisms. So a lot of my work has been on this, most recently looking at factors within that agricultural pathway. So I'm not going to go into the statistical details, but I just want you to see the end results. So taking those pathway characteristics, so nothing about the organism itself, just the pathway characteristics, and then looking at what influences what is found. Don't worry too much. First, year of import. So time is a big factor in what we actually intercept. And the reason this is difficult is because it makes predictions very difficult. So we know that things are going to change year on year. Therefore, it's very difficult to use historical data to predict what's going to happen next year. The size of the inspected sample makes a difference, which is sort of intuitive. The more we inspect, the more we're going to find. And then total volume of imports, same type of thing. What I also did was develop prediction models. So although predictions are really, really difficult in biosecurity, if I use these models and I use those influential variables to target inspection, so instead of taking a random sample of everything, we target the ones that we know are more likely to have contaminants on. And at low sample volumes, there's around a 40% improvement. So basically that's at 10%. So if we inspect only 10% of each sample, we'll still improve our detections by 40%. So we're working less, but getting better results out of it. This is my last slide, I promise. Okay, so at the end, what can we do? First, to focus on the pathway. We need to have more pathway risk assessments that all our other assessments feed into, and to manage pathways as a whole, rather than having most of our legislation directed at individual species. An idiographic approach, which basically means that different approaches for different problems. Each pathway needs to be looked at differently, and we can't just expect that because one thing worked for one commodity, it'll work the same for another. Thirdly, invest in people. I've worked in quarantine for many years, and there are many, many really good biosecurity inspectors and personnel. There are, unfortunately, it is government. There are also a few that aren't interested. And honestly, the difference that a well-trained, well-motivated person makes in how good your system is cannot be overemphasized. So more than anything, we need to invest in people. And it is that. Thank you.